All right, this is our last unit. Can you guys believe it that we're at the end? I mean, we have 4 more times together. That's it, and the semester's done, and then it's the final. That went lightning fast, in my opinion. This one's kind of cool from a unit standpoint because it pulls everything together at the end. If we recap what we've covered this semester, we started out with chemistry. And you guys are like, oh my gosh. And basic bio, you're like, oh. Right? And we talked about diffusion and osmosis, that bad word that starts with an O. And then we talked about active transport and phospholipid bilayers. You're like, oh, please don't remind me. But hopefully now when you look back, you say, okay, well, it does make sense that we learned about all that before we took this class. Because all of those parts are kind of key to understand what we're doing. Okay? Because if you don't understand the phospholipid bilayer, it's hard to think about how ions are going to move across or not move across the membrane. And if you don't think about the phospholipid bilayer, the transmembrane protein that has a channel, you have no really way to think about how to move a molecule across it. Right? And that was important for action potentials. And action potentials, we covered how they move and how they transport a signal, an electrical signal, so that you can make something happen downstream. And that might be like a muscle or a gland. We also talked about sensory, but we talked about it more generically or generally. And now we're going to talk about sensory in a more specific fashion. So we're going to cover the general senses as we kind of get into the special senses. And then we'll compare and contrast general versus special. And then at the end of the semester, the last couple of lectures, we're going to be here in our individual special senses. And those are all located in the head. And that's where you ended the semester in the lab with some of your dissections, with the sheep eye dissections, and looking at some of the lab activities on hearing tests, right, and taste tests. Did you guys do the taste tests in the lab? Okay. Did anybody do did you guys do the cut up the foods and then pair them with your lab partner?
No.
Anybody do that? Nobody did that, huh? Okay. You guys need to ask your instructor why I didn't do that. That one's cool. You guys, I'll explain it to you. You can do it at home, okay? You buy a raw apple, okay? Because the cooked ones in the grocery store are really hard to find. So buy a raw one. Get a raw potato, okay? Peel them both. Okay, I know that was a little sarcastic, but I thought it was kind of funny. Peel them both, okay? And then cut them into small little cubes. So it's just the flesh of you probably want to put them in separate bowls and label the bowls, okay? And then go to a roommate, have them close their eyes and plug their nose, okay? Actually, you don't even have to have them close their eyes. Just plug their nose and see if they can tell the difference between a raw apple and a raw potato. And I bet, they might, it's a 50-50 chance, right? So give them do a little experiment, give them 10 tries. They won't have to have dinner, they'll be so full. And see how they do out of 10 tries. My guess is they don't do very well. Because if you can't smell it, the texture is very similar. Okay? That's an activity you can do in lab. And we should have it all set up there. So ask your instructor, where's my apple, man? Where's my potato? Okay. So we're not there yet. We're going to special senses, and we're in general senses. And here's where we're going to kind of pull things together, because our sensory information, and a lot of times up until this point, much of the semester is focused on motor. We have a little bit of it focused on sensory information, like things come in the back part of the brain, from the posterior aspect with sensory, and then motor is towards the front. We talked about the dorsal root ganglion and information coming in from the posterior aspect with sensory and going up to the different parts. But most of the semester has been focused on doing something with the signal, like motor. Sweating when it was the skin, vasodilating, moving, walking, contracting a muscle, relaxing a muscle. So now we're going to really hone in on the sensory side of things. And the sensory side of things is critical. It's absolutely critical to us as a species. It's so critical that we've actually seen some really horrific results in various experimental models. Okay? So, I mean, some of these will disturb, some of you, but they've happened. There have been developmental studies in animals where they've restricted touch by a newborn mammal, okay, from the mother or from any other mammalian influence, okay? And they've developed psychological issues that are measurable, okay? So this concept of human touch is absolutely critical to our development, okay? And so when you think about a young infant or a young child not receiving enough of that nurturing, okay, or just confirmation that the world around them is okay, and sometimes it comes with just a hug or a high five or a pat on the head, and these are all integrating circuitry within our brains. Okay? We've seen experiments published, and these are many years ago. They don't do as many of these studies anymore because there's a lot of committees now that won't approve them. But they've restricted visual input in young mammals, okay, and animals. Okay, these weren't human studies. Okay, and these were organisms like us that had binocular vision, and by restricting visual input from one eye, you would have abnormal brain development on both sides of the brain because of the hemidesiccation that takes place. You guys remember that word? That's where visual input from the left eye goes to the right occipital lobe, but it also partly goes to the left side as well. And so you have the right occipital lobe, if you block the left eye visual input, it develops abnormally small, and so does the left side as well. Okay? So we know that the integrity of our intellectual capability or function is critically dependent upon sensory input with our surroundings. Information that are communicated by these sensory organs, many times we're not even aware of them. What's your pH level right now of your blood? Any idea? I mean, think about it. It's probably 7.1. You'd be on the floor, dude. I guess it's like 7.41. That's what I'm guessing. I'm just kidding. How many of you know what your pH is right now? Just by thinking. Of course you don't. How many know what your glucose level is if you're not wearing a monitor? Okay, those of you that are cheaters, don't look at your little device. Okay. So glucose levels, pH levels, oxygen levels. We're getting sensory input about those particular pieces of our environment. Many of them are internal environment, without us even consciously being aware of it. Our temperature. Now you say, wait a second. Well, at some point, your body temperature becomes consciously aware because it gets to a set point where you're like, I'm a little cold. Or, gosh, it's really warm in here and I'm sweating like a pig. Okay? So there is a point at which, you might even say, you know what, I just don't feel right. I've been running all day outside or working in this hot sun, and the only thing I've had to drink is water. I haven't had anything to eat. I skipped lunch. I've been out here for 8 hours. I think I'm dehydrated, and I have a feeling my electrolyte levels are low. Right? Now, you're consciously aware of that. I guarantee you that's true. But you just knew that because you started feeling either irritable or tired, fatigued, a little grouchy. Okay? Sounds like a Snickers commercial, doesn't it? So these sense organs, like blood pressure, body temp, and muscle tension, like, for example, the muscle spindle or the Golgi tendon, you don't consciously know that information when it's happening, but it's coming to your central nervous system all the time. It's telling your body where your arm is, where your leg is, how far away the floor is, right? Because you're getting feedback about your surroundings all over the place. You get up in the middle of the night, get a drink of water, or check the Christmas lights, or go use the restroom, and your bedroom is dark, and you're kind of feeling your way around your bedroom. You're getting sensory information about where stuff is, so you don't run into it. These organs, these sensory organs, utilize somatic and visceral reflexes, that should be familiar terminology, that are critical to our homeostatic survival. So if we flash back to the beginning of the semester, as we kind of wrap up the end of the semester, we're going to be pointing to this structure here. For 100 bonus points, just kidding. Who can tell me what this is? I'm surprised I didn't have like 5 people run up here, tackle the podium. Me first. This is skin. Very good. And what is this structure right here? This is a hint right here. This big M. What is it? A Meissner's corpuscle. You guys remember that from lab?
Yeah.
Vaguely, right? Well, this Meissner's corpuscle sits right between the interface of the epidermis and the dermis. It's in the dermis, right? This is a dermal papilla that's coming up, and these are the red apex that are coming down. Right? They fit together kind of like this for the structural integrity. If that didn't exist, every time you brushed up against the podium, a piece of skin would slough off because there wouldn't be enough structural interlocking between your epidermis and your dermis. And this is giving you information about pressure, right? Those Meissner's corpuscles are giving you information about pressure. And it's light pressure, right? Light pressure because it's so superficial. So those are the ones that tell you I'm wearing a long sleeve shirt today. Rob, you put on a long sleeve shirt today because I can feel the shirt sliding across my arm, right? Or it's telling you I forgot to put on my watch. How many of you can't really function if you don't put your watch on? And you know that your watch isn't on because you can tell, right? So these are the Meissner's corpuscles that are giving you feedback about your surroundings, okay, where things are in your environment. So the receptor is a structure that detects a stimulus. It has nerve endings within it that are going to fire, and it's going to send a signal back to the central nervous system and give you that stimulus to say, okay, I'm getting information about my watch not being on my left wrist. Okay? I have to turn the car around and go back right now. Okay? The sense organs usually are not just a nerve ending. It has tissue surrounding it. In some cases, we encapsulate it. We encapsulate it to enhance the response. And so the Meissner's corpuscle is an example of one that has a capsule around it. That capsule helps to confine that signal. The properties and the types of sensory receptors that we're going to go through are going to have different attributes. Like, for example, the receptor itself is a structure that's specialized to detect the response. Some of them are bare nerve endings. Some of them are true sense organs that have tissue around it. In other cases, they have epithelium or even muscle or connective tissue, like in the case of the Golgi tendon or the muscle spindle. Then another property is they transduce the signal. So they convert from one form of energy to a second form of energy. Because the first form of energy might actually be mechanical. Then the second form of energy is going to be electrical, right, with ions flowing. Because this mechanical touch doesn't make it to the brain. It takes that mechanical signal, it converts it to an electrical signal, and it sends it to the brain into the part of the post-central gyrus that corresponds to that part of the body. And then you get feedback of somebody's tapping me on the arm. I'm going to turn to see what they need. Okay? This transduction capability is a fundamental concept that exists for all transducers. Like, we use transducers all day long. I'm using a transducer right now to actually amplify my voice. Right? You probably used some sort of transducer to get here. What form of transducer did you use to arrive here today? Think about it. Let's hear some of them. What's that? A longboard. Very good. Okay. What's that? Your leg muscles. Very similar to the longboard, right? You're taking biological energy and you're transferring it into kinetic energy. Okay. What else? Some of you weren't as active to get here. Car. Okay. So gasoline engine, right? You're taking a fossil fuel, you're combusting it, and you're harnessing the energy out of it. So all these sensory organs are transducers, like what we utilize on a daily basis. So this light bulb above me is a transducer because it's taking electrical energy and is putting it into light energy. Right? So that's all the transducers, taking one form of energy, converting it into a second form of energy. And then a receptor potential. So we know what this word potential means now because we know that it's an electrical stimulus that is brought on by some sort of initiator. And it results in either a chemical release of a neurotransmitter or it results in action potentials, electrical signals. And they ultimately make their way to the brain, the central nervous system, for us to do something. And then last but not least, these are the 4 properties of a sensory receptor. Sensation. You have a subjective awareness of the stimulus. Right? A subjective awareness of the stimulus. So the receptor detects the stimulus. It transduces the signal from one form of energy to another. It brings on a potential so that ultimately it can make it into the central nervous system, go to the brain. And then last but not least, now there's a conscious awareness of what just took place. Many of these signals, you don't consciously become aware of them because they're filtered at the level of the brainstem, where a lot of the autonomic activity happens. Remember when we talked about central nervous system, one of the axis of orientations was sensory towards the back, motor towards the front, left versus right side, and then what was top down? Tell me what the top down was. How was the brain organized from top to down as far as functions? Conscious and unconscious are higher activity function up top. Like your intellect, your personality is in your forebrain. In your brainstem is where you monitor your heart rate or your respirations. Things that happen you don't have to think about. Language centers are up here. Okay? Whether or not you're beating at 60 beats a minute or 120 beats a minute is happening at the brainstem. Okay? So the brainstem filters out a lot of the information that comes in that you don't need to be focused on at a conscious level. Here's what I want you to do. I want you to take some time right now, and I want you to fill out this table. If you didn't print it out, you can just make a table on your sheet of paper. Okay? I want you to talk about the differences on the table between the general senses and the special senses. Give me some characteristics or examples on this first row. And then on the second row, give me locations of where you find these specific organs.
It's this one. So when
How are you guys doing? Let's start with general senses, because we've covered these before. So, shout out some characteristics of general senses. What's that? Characteristics. Give me some characteristics. Widespread. Okay? Actually, that might be location. I'd put that down here. It's a characteristic though too. So they're located all over the body and widespread. So that's that bottom box. What are some characteristics? Are they complex or simple?
Simple.
They're simple. Okay? They're very, very simple. In some cases, they're just a bare dendrite. It doesn't get much more simple than that. I mean, if you're in a bar fight and you said, stop being a bare dendrite. I mean, that'd be like ultimate insult. Okay? You'd probably get punched. You'd have to explain it maybe, but after that. Okay? Bare dendrites, very simple. Give me some examples. That might be easy. Examples of general senses. Is that a volunteer? No. Okay. Sorry. Temperature. Thank you. Right? How many of you have a thermometer? Anywhere? On your backpack or at your house? Okay? It's pretty simple, right? It's like some kind of liquid inside a tube. It goes up. You're like, dude, mine's digital. Okay, fine. It's a digital display. Very simple. What else? Temperature.
pH level.
pH levels. Very good. Pressure. How about one that we covered earlier? The example I used was what? A Meissner's corpuscle was a type of pressure receptor, right? Light touch. So these are all in this upper left quadrant. Okay, let's go over to special senses. Give me some characteristics. We're comparing and contrasting. You haven't figured out the activity. So,
Complex.
special senses, contrast. So what are the characteristics of special senses?
Complex.
They're complex. I mean, wait until we get into vision, right? And hearing. You're going to think, wow, this is very complex in comparison to the general senses. So, there's two examples right there, right? Vision, hearing. How many are there? Special senses? 5. You have a sixth sense, then you just watch the movie. That's all that happened. You might have a sixth sense, okay? So that's where that nomenclature comes from. Like, oh, you have a sixth sense, like you can tell that something's about to happen, right? That's what that's getting at. There's 5 special senses.
Taste,
Taste,
smell,
otherwise known as gustation.
Olfactory.
Olfaction, otherwise known as smell. That's the technical term for it. Other way around. Just kidding.
Olfactory.
Right? We got 4. We have vision, we have taste, we have smell, right? We have hearing. There's one we're missing. Equilibrium, otherwise known as balance.
Oh.
Now, where are those located?
in your head.
They're all found in the head. Right? None of the special senses are found outside of the head. You know, it's not like you're like, right? You're not like smelling your friends, right? Or tasting, you know, their sleeve, or hey, that looks yummy. Can I have a bite? Right? You know. So it's all with your special senses located in your head. It tastes good today. All right, next activity. I want you guys to partner up. Some of you that are in a row all by yourself, you might have to turn around. I want you guys to give each other an example of a transducer. Maybe pick one that we didn't cover. Pick one that we didn't cover. This is kind of an important mechanistic understanding of how sensations work.
What about the ones that feel like someone's beneath you? What would that be? Like if you feel like someone's following you, someone's beneath you, what type of sense would that be?
We just have like a very feeling of someone behind you. Yeah, it's just, you're probably hearing something, right? Or maybe you saw something move. You know, like in the movies, you're like, I think someone's following me, right? They probably got a glimpse of someone going around a corner, or they heard something. You know, they're a little bit paranoid anyways, and somebody might be there. So, I mean, that's just, you know, intuition. I mean, you're aware of what's going on, and you're paying close attention to everything that's happening. You're trying to pick up clues. your brain is putting the dots together.
Let's hear some of your transducer examples. Battery, nicely done. That's a really good one. Battery. How many of you have a cell phone? Right? Oh, 3. Yeah, so the 3 of you that have a battery, on the way out, would you be it? I have one too, so we'll cover all 4 exits, and we'll let the rest of your colleagues touch our cell phones so they can see what they look like. It's a giant transducer, okay? What's that?
Capacitor.
A capacitor, okay? What else?
Frets on a guitar.
Frets on a guitar. Very good. A nuclear reactor. Very good. Iron Man suit. Sweet. Okay. Absolutely. Okay. What else? What about the picture?
Solar panel.
Solar panel. Okay. Longboard. Right? Whether it's a skateboard or a surfboard, right? Huh? Oh, a lawnmower. Yes, a lawnmower. Yes, a lawnmower. Anything else? Radio. Nicely done.
Oh, yeah, no.
A tree. Okay, let's use a bio example first. Trying it out loud. Right? How about plants? Right? A flowering plant. How about us? How about the sun? Trying out loud, right? All of these are types of transducers. Okay, bless you. So here's why I'm doing these silly activities is we're going to get into the mechanisms, and it's really easy for students to get bogged down in the details and get lost. And they're like, I don't really, what's the whole point, dude? I mean, like, so, you know, this G protein goes to that one, and this mechanism goes here, this pathway leads to here, and you see all of a sudden. Okay, like, but I don't understand how you see. So the point that I want to drive home in the introductory lecture is, it's all about taking one form of energy and converting it into a different form of energy. Ultimately, you have to send the signal to the brain for you to detect it. So all the sensory receptors are doing are converting energy forms so that you can interact with your external environment and your internal environment. Okay, that's all that's going on. That's how all of this is working, is you're converting one form of energy into another. And as an organism, it's all biology. Everything in us is a biological type of transducer. But there's lots of examples that you interact with on a regular day, and they don't seem to be all that complicated. But for some reason, when students get to this section, they're like, I just don't get like hide and seek. Okay, like we've been talking about it for 2 weeks. I don't know, I don't understand. Yeah, like I just open my eyes, it's there, you know. It's like you're converting signals, okay? So light energy is going to stimulate a sensory receptor. Stay with me, guys. And that sensory receptor is going to be sensitive to light energy. And when the light hits it, it's going to cause some sort of reaction to happen on that receptor. That's going to probably either trigger a mechanism or open a channel and allow for ions to move. You guys with me? If ions move, can you take it from there? I hope so. You get an electrochemical gradient, you get a signal, that signal goes down a nerve to the brain, to that specific lobe, you're like, boom, I see. Okay? So I'm trying to give you the big picture. I know it sounds a little corny, but if I don't take the time to do it, a lot of you just memorize all the answers on the slides, and then next semester in 202, you completely have forgotten everything there was about special and general senses. They're just transducers, guys. That's all they are. So if we look at the general properties of a receptor, we've got 4: modality, location, intensity, and duration. The modality is, what type of stimulus is it? Is it light? Is it a photoreceptor? Okay? Is it taste or olfaction, which are going to be chemicals? You're going to respond to specific chemicals. There is a receptor on your tongue, inside your tongue, okay? And that receptor that specifically recognizes salt is going to bind sodium, and it's going to trigger an action potential, and then the food tastes salty. If you have a receptor in your nasal epithelium that binds a specific chemical molecule that corresponds to the active in lavender, then you smell lavender. Okay? That's modality. Location. This is encoded by nerve fibers that are issuing the signals to the brain. So location, obviously nasal epithelium, or the tongue, or the skin. Intensity. Which fibers are sending it, how many are doing it, and how fast they're firing. Right? So some of you know that, there are folks who walk in the room like, oh, they smell nice. There are others that walk in the room, you're like, they smell too nice. Like they should have not had, maybe only used 3 squirts of cologne instead of 12. Okay? So you can increase the signal. Now, on Thursday, when we come back, we're going to talk about pain. Okay? And I'm going to ask a couple volunteers. We got a little, I'm just kidding. But the pain is a sensation. It's a sensory input that's coming in. And so you know from an example of pain receptor that there is different intensities of pain, right? There's different intensities of pain that are low levels of pain or that are intense levels of pain. And then last but not least is duration. How long the stimulus actually lasts. So if the stimulus lasts a tremendous long amount of time, then you may actually develop a sensitivity to it and you may lose that signal. All right, the last thing that we're going to do today is I want to talk about receptive field. And we got a little demonstration, okay? And then this is it. So I want you to turn to your neighbor. You're going to partner up. But first, I'm going to get a volunteer. Would you come up and help me? Okay, actually, you know what? I need someone that has not a sweatshirt on. I need someone with just a t-shirt. Okay, how about you? Come on up here. Perfect. Okay. All right, sweet. What's your name? Mitchell. All right, this is Mitchell. Let's give Mitchell a round of applause. So we're going to demonstrate with Mitchell. We're going to demonstrate this example here. So he's going to take his shirt off. I'm just kidding. Just kidding. And I'm going to substitute a scalpel blade for that protractor. I'm just kidding. And on the back of the scapula, so turn this way. On the back of the scapula right here, right, we've got what we call a large receptive field. So if you look at the screen, it's one neuron that's going to a large area. And so the ability to discriminate information on the scapula is not as precise as it is on the hands. Okay, so I need you to face this way, okay? This isn't like some crazy trust exercise. I'm going to have you to fall backwards. I'm going to catch you, okay? I need you to close your eyes, though, because I don't, in this day and age, I don't know if you got like cameras everywhere. All right, so I want you to tell me how many fingers are touching this receptive field, okay? 1, okay. How about now? 1 again, okay. How about now? My whole hand. Okay, there you go. You did a great job. You were 33.3%. Okay. All right, no, we're not done. We're not done. Just come up here. Okay, now you're facing me. You're closing your eyes. Okay, again, nothing weird. But I need you to put your hands up. Okay, hands up. Okay. So now, how many am I doing?
That's 1.
That's definitely 1. It's not. Okay. How about now? 2. Look at this. He's 100%.
3.
Wow. Let's get it for Mitchell. All right. So why don't you guys do that with yourselves? Okay. Right now. Do it real quick. I want to hear laughter. So is it kind of working for you as well? So you get the difference? A small receptive field on your hand, you know, you have more neurons going to that same area to give you more discrimination. Okay, it's just two different designs on how it could be organized depending upon where it is on the body. Okay, I'll see you guys on Thursday.
I know.